Okay, welcome everybody. We're going to give it about a minute for people to file in before we get started. Welcome. We will get going at about a minute after the hour. Thanks for coming. All right, I've got 501 or 201, depending which coast you're on. Welcome, everybody. My name is Bob O'Sullivan. I am the Associate Publisher of Test Prep at McGraw-Hill. Welcome to our 5 Steps to a 5 webinar for AP Biology. We're about 6 or so weeks out from test day. So we're going to use this session to prepare you and give you some inside tips and information of the best way to spend your time between now and test day. Before we get started and I hand it over to our presenters, I would just like to cover a couple of housekeeping tips. Can you hit that slide, Kelsey, please? To begin, if you're a teacher or a student, we welcome you. Please do put the word out after this. It's available to both teachers and students, but it is primarily a student-focused review session, just so that's clear up top. In the beginning, before we started these things, we wanted to be pretty strict about questions and waiting till the end and using only the Q&A feature. What we found from doing a couple of these is that it's more organic, it's more engaging and fun. If you have a question while the presentation is going on, please just use either the chat or the Q&A feature at the bottom of your screen. Ask the question and we'll answer it as we go through. If you get in there and somebody's got the same question you've already that's on your mind, hit the thumbs up icon. That way we know it's important. You as participants should not be able to see one another, but you should be able to see me, Kelsey Burris, and Mark Anestis, our 2 presenters today. You should be able to see and hear all 3 of us. And then we've got Anya and Simone behind the scenes on the McGraw-Hill side, helping make this thing work. And you should be able to hear them, but you will not be able to see one another. Importantly, no student data is being collected. I realize that's a big deal for educators and for students. There's no risk of that. We're not collecting any names or any data. Don't worry about it. So that's not an issue. Before we get into it, I just want to remind everybody, we did design this to be an interactive session. I don't think there's too many questions that we're going to hit polling features with, but do feel free to engage with us. Again, it makes it better, makes it, more rewarding for everybody, both the presenters, students, and the teachers. So do participate. I would urge you to participate as the opportunity comes up. And the last point is the most important, that this session is being recorded. The recording will be available tomorrow, and that's your bit.ly. That's your URL at the bottom. I'll give you a couple seconds to take a picture of that with your phone. It's a 90-minute session. That's the first question. Thank you for asking. We're scheduled for 90 minutes. It may not go that long. But the recording of what we do here will be available at that URL, that bit.ly, by tomorrow. Again, put the word out there. We'd like to get as many eyeballs as we can on this for AP students. See if we can help people out before the test. So with that, Kelsey, you want to go to the next slide? I'm going to turn it over to Kelsey Burris and Mark Anestis. I'm going to have them introduce themselves and then we're off. Thank you. Over to you, Kelsey or Mark.
I'll start real quick since I'm less important today. I'm Mark Anestis. I started writing this book ages ago, and over the years, it's been modified and then modified again and then modified again as the AP curriculum has shifted. And Kelsey has stepped on the scene and helped shape this book into the beauty that it is today. And he's going to be running the show. I'm an SAT, ACT, AP tutor. And, you know, I think he does a great job with the book and the presentation. And he's going to walk us through what you guys need to walk through to get yourselves ready.
Thank you, Mark. So my name's Kelsey Burris. I'm an AP biology teacher at Union High School in Camas, Washington, which is a suburb of Portland, Oregon. I've been doing it for over 23 years now. And I'm an AP biology consultant. And I'm going to take some of the knowledge that I've gained over the years to help you try to prepare for the exam. If there's no other questions, we're going to go ahead and dive into the presentation. So our first order of business is we're going to talk about the exam itself. So it's very important that you understand the structure of the exam. So for the AP exam, there's two sections to it. There is a multiple choice section, which consists of 60 multiple choice questions. It carries a weight of 50%, and you'll have 90 minutes to complete that part of the exam. Then it is followed by a free response section, which includes 6 questions. 50% of the exam is the free response questions, and it's 90 minutes. We will dive into the structure a little bit more. As far as the units of the course, so these units might look familiar to you, or they might be named a little differently depending on your teacher. It doesn't necessarily have to be in this order, but if you take a look at this, so these are the ones that are laid out by the College Board. So you have the chemistry of life, which is technically like biochemistry. That's between 8 and 11%. You have cell structure and function, which is the organelles of the cell transport, that's 10 to 13%. Cell energetics, which is your enzymes, your cellular respiration, photosynthesis, that's 12 to 16%. Unit 4 is cell communication and the cell cycle, which includes mitosis, that's 10 to 15%. And then finally, you have Unit 5, which is heredity, which a lot of people refer to as genetics, that's 8 to 11%. You have gene expression and regulation, so that's your central dogma. So that's DNA replication, transcription, translation, and then eukaryotic and prokaryotic gene regulation, that's 12 to 16%. You have Unit 7, so in 5 steps to 5, we refer to it as evolution. It might be referred to as natural selection. That's 13 to 20%. And then finally, ecology, which is 10 to 15% of the course. The next thing that is important for you to understand is the importance of the science practices on the exam. The science practices are the things that you're going to be asked to show your content knowledge on the exam. So each of these science practices are actually better explained in 5 steps to a 5 or the AP biology course and exam description. So science practice 1 is concept explanation, and about 25 to 33% of the multiple choice will involve that science practice. Question 1, 2, 3, 4, and 5 of the FRQ sections will include that. So the way you can think about science practice 1, it's really content knowledge. All right, so it's asking you content. Science practice 2 is visual representation. So you're going to use a representation, a model, a figure, a diagram, a picture, or something to show what you know. And that's about 16 to 24% of the multiple choice section. And free response question number 5 really focuses on science practice 2, excuse me. Science practice 3 is questions and methods, and that's literally experimental design. So we're talking about everything from experimental variables such as independent variables. That's about 8 to 14%. Question 1 and 3 on the FRQ. Question number science practice 4, representing and describing data. So that's graphing and interpreting graphs. And you'll find that in question 2 and 6. Question 5 is statistics and data analysis. Again, 8 to 14%. And questions, FRQ questions 1, 2, and 6 will involve that science practice. And then finally, science practice 6, argumentation. 20 to 26% of the multiple choice will be asked you to do that. And questions, you'll notice 1, 2, 3, 4, and 6 have some component of argumentation in it. So now we're going to just kind of talk about overall this idea of AP exam tips, okay? So there's a saying that we like to use, which is stay calm and test on. So sometimes when you're taking AP exams, you will start letting the last question impact your next question. So what we want to do is make sure you stay focused. So here's some tips for you. So make sure you monitor your time appropriately through the exam. You want to ensure that you do not spend too much time on one question and not have enough time on the other questions. Now, most of the questions on the exam tell a story using scenarios. And this is really important because you do need to have content understanding to understand these scenarios. And that's kind of where like 5 steps of 5 would come into play, right? Where you're making sure you have that content knowledge you have. And so when you're looking at the story or these scenarios, you got to look for the biology in there. Is it talking about cell transport? Is it talking about cellular respiration? And the thing about it is that you might not have any prior knowledge of the scenario, which is really, really important because the biology is inside there. You're just reading something you might not have seen before. You might be looking at an experiment that you've never seen. And it's important to focus and find the biology in that story. And don't panic when you come across unfamiliar content. A lot of times students will start to stress the fact that they might be think the questions about, let's say, the immune system, which is not on the exam anymore, but they're just using it as the scenario, and there's biology inside of there that you need to look for. So make sure you calm down and focus on the questions you know. And remember this, and Mark would be able to add to this, there is no penalty for guessing anymore. Many, many years ago, you used to get penalized for guessing. It was the same thing on the SATs and all that stuff. That's old news for us. So do make sure that you stay calm and that you test on when you're going through the exam. So for the multiple choice section, so the section, like we were saying, includes 60 multiple choice questions. And there's 2 types of questions. There'll be individual single questions that are not connected to any of the other questions on the exam. And then they have sets of questions that will refer to the same diagram or data presentation. So, and those usually sit in question sets of 3, 4 to 5 questions. And the biggest thing that you got to remember is that the multiple choice section is 90 minutes long. You have 60 questions, so you need to average roughly around 90 seconds per question. So whenever you're doing any practice on the exam, you got to make sure that you are practicing at that clip, so you get accustomed to the time. Most students who practice with time limits and stuff, the AP exam is much easier for them when they're working on that time, those time limits. Now, your questions will test your ability to do some things, right? So explain biological concepts, processes, models, analyze diagrams, flow charts, and other visual representations, okay? Remember, those could be pictures. They could be things that are unfamiliar with you, but you gotta find the biology inside them. They might ask you to use scientific method, right? In terms of the experimental design. What variable is that? Identify a testable question. You might be asked to perform mathematical calculations of some sort. And you might be asked to support a scientific claim using evidence given to you in a scenario. So let's do our first question here. So what I would like you to do is take a second and analyze this question, and then use the poll to answer your question. We'll give you about a minute to 90 seconds to do this.
For those of you guys attending live, it's easy to click and drag. If the polling is blocking this question itself, just click and drag it over to the side.
All right, as you're kind of finishing up here. So on this, the answer is B. Sorry, there we go. So the answer is autosomal recessive, which quite a few got that right. What the reason I threw this on this presentation is the fact that pedigrees are really important for you to be able to analyze and to be able to understand. They are usually very common on the exam where you're able to ask a lot of different questions. So when we're talking about science practice 2, using a visualization or a model of some sort, a pedigree is an example of that. The reason that the answer is B on this question is it is not autosomal dominant, because in order for the second generation on the left to have 2 individuals with the condition, one parent would need to display the condition as well. It is probably not sex-linked because it appears as often in females as in males. So autosomal recessive seems to be the best fit for the disease. So let's go ahead and jump into another pedigree question using the same pedigree, right? So we're practicing this idea of a question set, right? So it's the same figure that you're going to be using, but now we're going to use a different science practice to answer a question. So we'll give you about 90 seconds to answer this next question. All righty, so it looks like we have the results up here. So our answer for this is D. All right, so one first needs to determine the probability that the person C is heterozygous. We know that person D is double recessive because she has the condition, so she's homozygous recessive. We know that the parents for person C must be heterozygous, both parents, because neither one of them has the condition, but they produce children with the condition. The probability of the person C being heterozygous is 2/3. Because it's a monohybrid cross of the parents, and since you know that he doesn't have the condition, he cannot be homozygous recessive, little b, little b. This leaves just 3 possible outcomes, 2 of which are heterozygous. A cross must then be done between the father, person C, and the mother, person D. The chance of their child being homozygous recessive is 50% or 1/2. This means that the chance of these 2 having a child with the condition is 2/3 times 1/2, or 1/3, or 33% in this case. All right, so let's try another question here. So on this question here, we're going to look at phylogenetic trees and a derived characteristic chart. So we'll give you a minute, 90 seconds here to interpret this graph, and then we'll go over it real quick. All right, excellent. So we have our responses. And on this case, the answer is actually C. And the reason it is C is that there are fewer nucleotide differences between species 1 and 2, indicating they would reside close to one another on the cladogram. The same holds true for species 3 and 5. There are a large number of differences between species 4 and all the others, which then leads to C being the most appropriate answer for this one. So we took a minute to look at pedigrees, and also phylogenetic trees are just two examples that a lot of students forget about when you're dealing with science practice 2, being a model that you have to be able to interpret.
Can I just, before we go to free response, can I just chime in on multiple choice, just strategy?
Yep.
As you talked about the timing, how important that is, and you have the 60 questions and the 90 minutes. And I just want you all to remember that you do not have to move sequentially in terms of spending your time. So if you come across a question that you know you struggle with, or you have no idea what it is, don't continue to work at that question until you figure it out. You can box that question, bubble something in, move on, get through the entire multiple choice section, so that at the end, when you're pressed for time, you're working on the questions that are the hardest for you. You're going to try and streamline it a little bit, so you're working on the hardest ones at the end, so you're getting as many points as you can and accumulating them, and leaving the hardest stuff for the end.
Excellent point. So we're going to segue that actually right into the FRQs, because we're going to use the same strategy when we talk about the free response questions. So when we speak about the free response section, there are 6 questions that have to do it, and you have 90 minutes for it. And to piggyback on what Mark said, a lot of students, they do number 1 first, followed by number 2, number 3, all the way to 6. And most years, the lowest performing question on the exam for FRQs are usually the last question, like in this case, question number 6. The reason is students don't get to it. So one strategy that you would want to use is do a quick overview of these 6 FRQs, and then number them from easiest to hardest, and then answer them in that order. So you might go 2, 4, 6, 3, 1, 4, or something like that. Okay. So when we look at the free response questions, you'll notice that question number 1 and 2 are 8 to 10 point questions, and you're going to average about 25 minutes on those, because they are considered the long FRQs. They're going to be longer in length. They're going to have more sections to them. The one thing that happened in 2019 on the AP exam, which was given in 2020 for the first time, is that the questions are somewhat standardized, where we know kind of what they're going to be asked asking in terms of science practices. So like question number 1 is interpreting and evaluating experimental results. Question number 2, same thing, but with graphing. 3, scientific investigations. 4, conceptual analysis. 5, analyzing a model, and then 6, analyzing data. Questions 3 through 6, excuse me, are what we refer to as the short answer ones. And these are 4-point questions, and you should average roughly 10 minutes on them. And when we put these time periods on, it doesn't mean that you can't work over, but what we like to say is like work up to that time, and then you can always come back to it. But what you don't want to do is spend 40 minutes on one question and then leave the other questions. There's only so many points you can get out of each question. So here's some tips for free response questions. Again, monitor your time appropriately, okay? And what's a challenge is some of you don't own regular watches and stuff like that, and they're not going to let you have your cell phone in there, which you live on, and most likely your Apple watches are not coming in. So you might have to, borrow a watch or something, because it does help your time in case you can't see the clock in the room wherever you're at. So do not spend too much time on one question, not have enough time for at least attempting all the questions. So we talk about this in because when we grade AP exams, which is get to the point. Do not rewrite the question. So you have to understand that when teachers are the ones that grade your FRQs, and you grade the same question for a week straight, and we know the question really, really well, so you're not going to trick them by rewriting the question as the answer. So what we want you to do is just ATP, which is answer the prompt and get to the point. So you want to do everything the prompt suggests. So for example, when we say just answer the prompt, what we're talking about, if it tells you to describe, make sure you describe. If it tells you to identify, make sure you identify. If it tells you to graph, you graph, okay? And write incomplete sentences, all right? Doesn't mean that you have to be, that you're going to be graded on your sentence structure, but it clears up your thoughts. Do not list things. Listing things indicate you only list a thing if someone asks you to do that in the question, which we're not asking you to do. So make sure you write incomplete sentences. Also, on the free response questions, this is to help the grader. Remember, the reader is trying to award you points, and you want to actually kind of make it easier for them, all right? So when you are writing these questions, you've got to think about these FRQs as being in separate sections. So you have section A, section B, they're separate questions you got to think about, right? It's not just one gigantic essay. So if you notice, we talk about free response questions. They're questions. And so question A, you write it and then skip a line, then write B, skip a line, write C, skip a line, write D, okay? Now, if you mess up, you don't have to erase, you just cross it out, you draw errors, we will find your answers, what you're after. So if you misspell something, you know what, just cross it out and move on there. So one of the things that that I work with my own students on is this idea of deconstructing free response questions. So we're going to talk through these steps. And remember when I was talking about these FRQ questions that you will have a scenario of some sort, and you got to find the biology in there. So the first thing we tell our students to do is examine any data and model present in the prompt. It might be a picture, it might be a graph, it might be, but those pictures and graphs are telling you a million things, okay? And what you got to ask yourself is, what do you see, and what does it mean? What biology is there? What is it talking about? Because that will actually help you in the question. Then we're going to ask you to read the prompt and look at the bold-faced terms. What are the task verbs, which we'll talk about a little bit in a little bit. And we're going to have you ask, what are you being asked to do? Are you being asked to define? Are you being asked to explain? What are you being asked to do? Next, that's when we're going to have you read the scenario and or investigation. And we're going to ask you to think and ask yourself, what biology concepts are present? Think big picture. Are they talking about cell transport? Are they talking about cell communication? Are they talking about natural selection? What are they talking about? And remember that most of these questions are they tell a story using a scenario. So look for the biology in that story. Do not get caught up on the scenario, okay? And then last, again, read the prompt. So if you notice, I keep talking about reading the prompt, and most importantly, read the prompt, because a lot of students, what happens when they're answering these questions, they just don't answer the question. So you're waiting for them to say it, and they're just not saying it because they're not doing what they're being asked to do in terms of their task verbs. So these task verbs are listed here, and you can find them in 5 Steps to a 5, and they're always in a bold print. So they're telling you what you need to do. They're put in bold print to help you to know what you need to do on the exam. And I want to bring to your attention a few of these, which actually layer on top of each other. And it's where students struggle when we're grading FRQs, is the idea of first, okay, so most students can identify, that's not a problem. But then what they do is they still identify when they're supposed to describe. Okay, describing is like, it wants you to say what is happening. But then there's another layer above that, which is explain, where students, when they see the word explain, they start describing. And you remember explaining, we want you to say how something is happening and why. Now you're bringing depth to it. And then the last one is justify. So when we talk about justify, it's talking about, so you're providing evidence, okay? And you, it might be like a lab scenario or something like that, and there's a claim, and you need to provide evidence to support, qualify, or defend the claim. And that just, what a lot of students do is they just repeat the evidence. Well, the evidence is in front of you. Yeah, you pull the evidence, but then you also have to provide some reasoning to explain how the evidence supports or qualifies the claim, which is, that's biology, okay? That's where you're kind of taking your content knowledge, which you can pull, if you use 5 steps to 5 as a review, you can pull that from your experience as you reviewed from the book.
Kelsey, just a quick time check. We just clicked by 30 minutes. So we're 30 minutes in.
Perfect.
Doing
Okay.
great. Thank you.
All right. So now we're going to talk about each of the FRQs. Okay. And the FRQs, they're kind of not standardized, but they are. We know what they're going to ask you in some form. Now, for question number 1, which is interpreting and evaluating experimental results. So part A is going to ask you in some fashion to describe and explain biological concepts, processes, or models in some fashion. Part B is going to ask you to identify experimental design procedures, which is science practice 3. So they'll ask you, like, maybe, you know, what are the variables? What is a posable question? What were the controls? Part C is going to have you analyze data. And then part D is they're going to ask you to make or justify predictions. Now, I do want to point out part B, where identify experimental design procedures. In AP Biology, there are no prescribed labs. There's not prescribed labs, okay? So whatever scenario they give to you, they're going to give you enough information for you to understand it. Remember, it's about the biology behind it, okay? And so you have to be able to make sure you look through the procedure, the prompt, make sure you look through the scenario and find that biology in lab experience. So whatever lab experience you had in your classrooms, you can take that experience, right? Because you were working on skills and working on experimental design, and then make sure you find that biology in that scenario, that investigation that they give you. For question for question 1, it's 8 to 10 points, and it's going to be just to add, it's going to be an authentic scenario. So it's going to be a real life experiment that you see, okay? And it's going to have a data in a table and or graph for you to interpret. All right. So for question number 2, you're going to see that it's very similar to question number 1, but in question number 2. You have graphing. So if you know it's part A, same thing. So you're going to describe and explain a biological concept, processes, and models for a couple, 1 to 2 points. But part B, you're going to construct a graph, plot, or chart using confidence or error bars, which we're going to talk about both of those in a little bit. You'll need to analyze data, just like question number 1, and then make a justified prediction. Now, I want to be clear, these questions can be structured in a different way. These are just an overarching idea of what they're going to ask you to do. All right. So those are the first 2 long ones. So the question number 1, question number 2, you should only spend 25 minutes on it. I do tell my students is like, you got to look at each of these. Part A as being its own little question, part B being its own little question, part C being its own little question, and that you can gain points at any point. It's about gaining points. That's what you're after. And I talk to my students about how it's additive grading, not subtractive. You all start at 0 and you need to gain points is what you need to do. So now we're going to talk about the 4 short ones. Okay. So for science practice, for FRQ number 3, it's a 4-pointer, and it presents a description of a lab investigation scenario. So again, this investigation is most likely will be something that you might not have done in class, but you got to look for the biology in there, and they're going to ask you to describe a concept, process, or models, identify experimental procedures, predict results, and then justify those predictions. FRQ number 4 is going to ask you to present it's going to present an authentic scenario, describing a biological phenomenon with a disruption. So something that causes it to change course. So part A, describing biological concepts. Part B, explaining these biological concepts. And then you got predict the cause or effect of a change in a biological system. So what would happen if the temperature changed or whatever it might be. And then you have to justify those predictions. So if you notice, when I kind of highlighted describe and explain and justify, you'll notice they're all over these things. Now, they might use different task verbs, but it is a very important thing to understand when you're writing these FRQs. Question number 5, analyze model and or visual representation is, again, another 4-point question, which, again, you shouldn't spend more than 10 minutes on. And it's going to be presented, it's going to present a description of an authentic scenario. And it's going to be accompanied by some visual model or representation. It might be a pedigree, it might be a picture of a cell, it might be all sorts of things, okay? And you're going to describe the characteristic, part A. Part B, you're going to have to explain it, the relationship in some way. Part C, you're going to represent relationships within a biological model. So it might be how you have to connect like transcription and translation to cell communication or something like that. And then part D, you're going to be explaining in some fashion. Okay, now again, I do want to reiterate that these task verbs might be a little different in the question, but it gives you the general gist of what the questions will be structured like. And then finally, FRQ, free response question number 6. You're going to look at a piece of data that's in a graph, table, or other visual representation, and you got to look for this biology in that data. And part A will ask you to describe, part B will ask you to describe the data some more, part C will ask you to use the data to evaluate, and then part D will ask you to explain how experimental results relate to biological principles, concepts, processes, or theories. So with that being said, that's the structure of the exam. And I took time to go over the structure of the exam because it is important for you to understand the structure of the exam when you're reviewing and understand that the need to practice these things. So where we're going next is like, I'm going to kind of like, we do not have time for me to go through content, right? That's not what this is for, but I'm going to highlight some things that I think are important. And some things that you might need to focus on, because in my 20+ years, my students, these are usually like the hallmarks. They might be good at one, but another one they struggle at. So the first thing I want to talk about is how everything is connected. Remember, everything in life is connected to each other, and that biology is no different to them. Okay, so one of the struggles that teaching AP biology all these years is it feels like you're on this course where nothing's connected until the very, very end, and all of a sudden everything's related to each other and you're making all these connections. And it's important at the end that you can make these connections. So when you're going back to review, you got to think about those things, and you got to think about the scaffolding of topics. You got to think about how the stuff from Unit 1, which is like your proteins and stuff like that, all play a role throughout all the other units throughout the course. And so when you're reviewing, it's not just reviewing about the topic in front of you, but also thinking about other topics that they connect to. So like, for example, when we talk about sickle cell anemia, which is an illustrative example in the course, and how sickle cell anemia can be connected to a whole bunch of things, right? It can be connected to protein structure. It can be connected to gene expression. It can be connected to mutations because it was a point mutation of the red blood cells that led to the sickle cell trait because of a missense mutation that leads to a change in protein and change in the amino acid structure, which leads to a change in the protein structure, and then you have your sickle cell. And then on top of that, right, so we're talking about, that's Unit 1. We're talking about Unit 6 right now, when we're talking about gene expression, right, mutations and all that. But then it goes even further, because you talk about this in Unit 7 when we're talking about heterozygous advantage and the fact that sickle cell is an autosomal recessive disorder. But when it's in areas with malaria, the heterozygous is has an advantage because they are able to, they have a little bit of sickle cell, but they're also resistant to malaria. So talking about scaffolding, now we're going to talk about the big three I refer to. These are the three topics that students struggle the most with. And when I talk about these overarching concepts, these are things that as you're preparing to review, you got to make sure that you kind of take a look at. So first one is unit 3, which is cellular energetics. And the things inside cellular energetics, you'll find enzymes, cellular respiration, and photosynthesis. And so make sure you take a look at that. Now, for cellular respiration and photosynthesis, do remember, you need to know, like I tell my students, know what goes in, what comes out, where it takes place. All right. The next of the big three I refer to is cell communication, unit 4, which is dealing with signal transduction pathway, which is a challenge for students. And then finally, unit 6, which is gene expression and gene regulation. Okay, if you notice, I did not put genetics in here. I put gene expression and gene regulation because it includes all the central dogma, right? DNA replication, transcription, translation, and then the 2 gene regulations, which is prokaryotes and eukaryotes. So do make sure you take a moment to focus on those. Now, the other things all connect back to it, but these are usually the big, some of the bigger arching concepts that students struggle with. Now, as far as some skills, okay, so graphing and error bars. So with graphing, I need to bring your attention to a couple things. So types. So you've got to be able to identify which type to use. X and Y graph, log Y graph, a histogram, a line graph, a dual Y, a box and whisker plots, and a pie graph, okay? So you might not have seen these in your classes, and so you got to make sure that you practice these. Now, on the exam, when we ask you to graph in FRQ number 2, part B, we're looking for these things. Orientation, right? Are they on the right, or the X and Y axis? The labeling, the units, proper scaling, plotting of the points, the type of graph used, and the trend line when appropriate. You will also be asked to interpret data, okay? So you got to be able to identify data points, trends and patterns and relationships between variables, because you will find graphs all over the exam, okay? And so you've got to be conscientious that you are able to take a look at that and interpret that. And then finally, confidence intervals and or error bars, all right? Which is a challenge for a lot of students, and you will see them on the exam. And when we're looking at that, we're looking at the idea of 95% confident intervals, so plus or minus 2 standard error of the mean, and from that to be able to determine the statistically significance of the data. So with confident intervals, so real quick, standard deviation. So when we're talking about standard deviation, remember that standard deviation determines the amount by which your values or data points typically differ from the mean value, and that the standard deviation determines the amount of variation in your data. Standard error infers how well the sample mean represents the true population mean, helps determine confidence in the data collected in a sample, and set the 95% confident intervals, which equals plus or minus 2 of the standard error, and be able to place error bars on the data sets. Okay, so you got to be able to put the error bars, which are in red here. Standard error is estimates the likely accuracy of a number based on sample size, while standard error of the mean indicates the size of the likely discrepancy compared to a larger population. So very similar in things. Most of the time, what you'll see on the exam is standard error of the mean. All right, so error bars, right? So I'm talking about that, and some of you are like, I don't graph, but there's a really important part about error bars that you have to be able to do, which is be able to interpret them. So looking at the red, you'll notice that we have two sets. We have the set on the left and the set on the right, in terms of error bars. And when the error bars do not overlap like this, then you can claim that the data is statistically significant. There's a difference between those data points. If the error bars do not overlap, like in the set on the right, you could not claim a statistically significant difference between those data points. So that mean could actually be the same. Next, so when we're talking about mathematics and statistics, okay, so you could bring a calculator into the exam, and it could be a scientific calculator, but it must not have any unapproved features or capabilities, okay? But your cell phone will not be the one you can use, okay? But all that, like I tell my students, a simple 4-function calculator that you can buy at the dollar store can solve any math that is on the exam, all right? It's not complicated. You will be given a formula sheet that has the equations on it, and you will be able to use it in both the multiple choice and the FRQ questions. So you don't have to memorize these questions, but formulas, but you do have to be able to use them. Now, the formulas, so we have the Hardy-Weinberg, which is from Unit 7. You have chi-square analysis, which you can use when you're dealing with count data, like in Unit 5 with mitosis, Unit 4 on mitosis, Unit 5 with genetics, Unit 8 with like animal behavior. You have water potential. You have a Simpson's diversity index, which is one of the newer equations that was added in 2019. And then you have rate and growth, which we're talking about population growth, exponential growth, logistical growth, and that's found actually in Unit 8 as well. So we're going to talk about a couple of these real quick, just as an overview of these equations. So with Hardy-Weinberg, you remember with Hardy-Weinberg, there's actually two equations. There's p squared plus 2pq plus q squared equals 1, and then there's p plus q equals 1. When we're talking about p squared plus 2pq plus q squared equals 1, we're talking about like an individual that has two alleles, okay? So you have the homozygous dominant, which is equal to the p squared. You have the well, that should, excuse me, that's a misprint. That should say heterozygous, okay? And then the key with this one, though, is to remember that this, both of these, p squared plus 2pq, are both contain the dominant phenotype, okay? And then you have q squared, which again, that should be, it's not homozygous dominant, that is homozygous recessive. So that's a typo there. Now, that one there is the recessive phenotype, and you can tell that one 100%, okay? So when you get a problem, whatever, q squared is the homozygous recessive, and it's the recessive phenotype. Now, p plus q equals 1, you're dealing with single alleles. So p is the dominant allele, okay? And q is the recessive allele. Now, the key to all these equations is to get to q. So if you can get to q, then you can find p, and then you can go back and find p squared plus 2pq, okay? All right, now conditions for Hardy-Weinberg equilibrium. So these are the things. So when we're talking about Hardy-Weinberg equilibrium, we're talking about the idea that things that will keep a population from evolving, okay? And there's 5 of them, and all 5 of these things have to be taking place at one time, at the same time, in order for a population to be evolving. And what they are is, one, large population, which means no genetic drift, no migration, which is no gene flow, no mutations, no natural selection, and then random mating. If any one of those things is taking place, then the population is most likely evolving. All right, so that being said, we are going to look at chi-square analysis, okay? So chi-square analysis is appropriate when you're doing count data, okay? And this equation is given to you. So that Greek looking symbol means sum of, O means observed, And then E means expected, okay? So anytime you have observed data and expected data, so like, for example, flipping a coin, you would expect, if you flipped it 10 times, you expect 5 heads and 5 tails. Well, that would be your expected ratio, where observed would be whatever you counted with it. One thing about the chi-square analysis, you do have to understand null hypothesis, which suggests that there's no significant relationship that exists between the observed or expected values. The differences are due to chance. And then degrees of freedom. So this chart is given to you, so you don't have to memorize it. But there's a couple things that you have to know about the chart, the p-value. So degrees of freedom equals the number of count groups minus 1. And the p-value we use is 0.05 or 95% confidence. So when your chi-square value, which is x squared, is less or equal to the p-value, you fail to reject your null hypothesis, which basically mean that any differences between your data points is just due to chance alone. When your chi-square value is more than the p-value, you reject your null hypothesis. So there's a reason that your data is different. All right, so next one here, water potential. So with water potential, so we're looking at the idea of pressure and solute potential. And this equation can be a challenge because there's two parts of the equation. So pressure and solute potential. Pressure potential is always 0 unless it'll be given to you. So like inside a plant cell, there's pressure potential, but in an animal cell, there is not, okay? So by that being said, a lot of times when you're looking at water potential, you have to solve for solute potential. So solute potential is a formula negative ICRT. I is the ionization constant. So for sucrose, it's 1, meaning it doesn't separate, right, when it's dissolved in water. C is your molar concentration. So you're going to need to pull that out of the question. R is the pressure constant. So it's always 0.0831. And then temperatures in Kelvin. So they will give you the temperature in Celsius, and then you'll need to transfer it by adding it to 273. You multiply all those together and you get your solute potential. And if pressure potential, if it's in an animal cell or an open container, water potential will equal solute potential. And when you're going to use these questions are really about when you're looking at hypertonic, isotonic, and hypotonic environments. So you're going to compare these water potentials inside and outside, and you're going to be asked which way is the water moving or another solution. All right. With that being said, we're going to talk a little bit about the AP exam review and like a review schedule, which we do lay out in the 5 steps to 5. So with the review schedule, there's a couple things that you need to figure out. Well, how much time do you have left, right? Every day and week goes by, you have a little bit less time, so you have to set a calendar, okay? Now, in 5 steps to 5, you will find that there is in chapter 2, how to plan your time. Now, if you're just starting now, that calendar will look a little different because a lot of times 5 steps to 5 is talking about whole semester or something like that. But if you're talking about 4 weeks, 3 weeks, 5 weeks, or whatever it is. So if you have 8 weeks to exam, you have about 1 unit per week. But if you only have 4, which you're getting too closely because the exam's on May 10th, you'll need to review 2 units per week. You need to determine your test readiness. Take a look at the diagnostic exams. You can look at chapter 4, where how you can approach each question type. And then ultimately commit to your plan. Use your 5 steps to 5 to gather that content knowledge, review the content knowledge you might have got in class or you missed in class, and start building that, because that content knowledge is going to be so important when you're reading these scenarios to pull the biology that's in there and to be able to apply your knowledge. And then ultimately practice, practice, practice. All right. So with that being said, let's do a quick debrief and summary of what we kind of went through here. So as we went through here, we looked at the structure of the 6 free response questions. We also looked at the multiple choice section. We looked at task verbs, so don't forget about the task verbs. We took a look at exam review, which, look at a plan, use the 5 steps to a 5. We looked at the free response question tips. So again, you notice I'm gonna bring these back up. So when you're looking at these free response questions, first examine the data models present in the prompt. Ask yourself what you see, what it means. Then read the prompt, look at the bold-faced words, right? The task verbs, what are you being asked to do? Next, read the scenario and or investigation. What biology concepts are present? Think big picture. Last, read the prompt again. And finally, remember, stay calm and test on. With that being said, I'm going to go ahead and pass it over to Bob, who is going to talk about the online prep course.
Thanks, Kelsey. Great job. All right, so I'm just going to spend maybe between 5 and 10 minutes introducing you guys to our online course. By attending this session, either live or in recording, you're getting access to what we used to call our cross-platform prep course. Now we just call it the online course. If you take a second to take a picture of the URL and the access code or jot it down, that's going to get you in. Really easy sign up. Don't worry about being contacted. You will need to enter a name and an email address to get in. We will send you exactly 1 email just to verify that you're in. You won't get any spam or anything from us after that. So no worries there. All right, so now you've got, we'll show you again at the end. You've got the URL and the code. Kelsey, if you can stop sharing so I can start sharing. Okay, can you see that? This dashboard? Anyone? You can see it. Good. All right, so the first thing you'll see, and I'll run through these a couple of features, but I will say up front, I really suggest you guys go in and check it out yourselves. The best way to see the resources that are in here and how useful it'll be in your test prep is to take the code and go in here yourself, but I'll show you a little bit. The first thing you'll see when you come in is that every time you come into the platform, you'll see this intro dashboard. I looked up while Kelsey was talking, I looked on the internet to see that the date of this year's test is May 10th. So that actually, it means that we're 43 days out today. So your custom study plan will update every time you come in. Today it's 43. A couple days from now, you'll be down to 40 days of studying. And that'll update every time you come in. The same with your study plan progress, your knowledge goals, your percentile rank, your average score on a practice, average score on the test, and then your strengths and weaknesses in each category. I am, haven't done this. I just logged in for the first time here today. So one of the cool things about this is that it will automatically update for you and you'll be able to see your progress every time you come back to the platform. Just a quick note, it's real easy to download on your phone too. So if you're working on your laptop in school and you're doing a practice test and get up to question 25, then you got to leave and jump on the bus. If you log in on your phone, it's going to pick up at question 25, right where you left off. No matter which device you're using, it's going to follow you and update. So it's a real easy situation. So that's your dashboard. That's the main thing that you'll see first. Then just to run through a couple of the features, you've got your study plan, which is everything that's in the 5 Steps to a 5 book, just broken down. You want to look at respiration. You want to focus on cellular energetics. Everything that's in the book is here. Is basically this section is basically a coursified version of the 5 Steps to a 5 book. You've got a game center in here, which is everything okay?
He fell on his wrist and was going to get an X-ray.
Who?
Well, looks funny.
Sorry, everybody. I'm having a little chaos at my house. My son is heading to the emergency room, but it's just his wrist. He's fine. I knew there was something going on. I kept looking out the window. So there's some games in here, some flashcards, pretty simple flashcard based games. That again, if you're on the bus or studying on the go, it's all your AP biology material that is available for you to do. There's a discussion board. I usually find these are, there's a couple of things in here. These are pretty rarely used. You can often see students back and forth. Hey, what did you get on number 12? Or did you think it should have been C and not D? That type of stuff. But the real value is going to come toward the bottom here. You've got your lessons all broken down. I just, while Kelsey was speaking, went through the heredity section. And as I went through, I just quickly self-graded myself. I know most of it. I'm grading myself as high, but it got down to some stuff on intermediate inheritance, other forms of inheritance, other things that I thought maybe I wasn't as confident on, sex determination, linkage, and gene mapping. Basically, this is a self-guided tool. So as you go through, if you rate yourself high or low, then you know next time you come back into the platform where you rated yourself low, that's you telling yourself, hey, I want to come back to this, or this is a topic I really think I should revisit as I get closer to the exam. So it's a really helpful way of going through all the content and weeding out the stuff that you think warrants more attention. The flashcards, I think, are the best part about these. You've got 772 AP biology flashcards in here. I think that might be a record. That might be the most of any subject. So again, if you're on the go, you're on the bus, you're anywhere, you just quickly, one click, you can get the definition. Again, did you get it right? No, kinda, or yes. You grade yourself. If it's a yes, you probably won't see it again. If you grade no or kinda, it's going to come back. Next time you come on, you'll see this again. So these are great. I mean, all this stuff is relevant to the test, relevant to things in AP biology you need to know here with one click. And again, one click. And super useful. Grade yourself as you go on, and again, it'll customize for your needs. In the practice, these are what do you got here? 10 or so sections broken down of practice questions. While Kelsey was talking, I ran through the heredity again, and I actually did pretty well. I got 43, not knowing anything about the AP biology. I got 43% of them correct. These you can go through and answer as you go along. You can either set it so that the answers pop up as you go through, and each question you'll see as you proceed, the answer will come up. This one you'll see on crossover frequencies. I didn't get it right. As I moved forward, it showed me my wrong answer and then the correct answer with the explanation right here. So yeah, these practice questions are broken down. You've got 14 on heredity, 11 on molecular genetics, everything. So a really useful set of questions and answer explanations as you go through. And lastly, definitely most importantly, I think we can agree, are full-length practice tests that are available to you. You've got the entire 3-hour test here 3 times, 3 different sets of questions. One's a diagnostic test you can take the first time you come in, see how you do. And the other one's got practice exam 1, practice exam 2. You'll sit, answer the whole thing in 3 hours. Even if you feel super confident getting closer to test day about your knowledge of AP Bio, we all agree, and I'm sure Mark and Kelsey would attest to this, taking at least one full-length exam is good practice for you. Sit, do the 3 hours, do it at least once all the way through. You know what you're getting into. You know what you're going to be going through on test day. So it's a really good tool. If nothing else, then just to prepare you for the exact number of questions you'll see on test day, for the exact amount of time you'll be given. And then, of course, it's in test mode. You won't be able to see the answers and explanations as you go through. You'll have to wait till the end to get those, and then it'll estimate a score for you. And that's it. That's the highlights. And then you've got a couple things over here. Your some access to personal information. You've got little awards. Congratulations, you've unlocked getting flipped. Little email, fun little messages you'll get from time to time. But that's pretty much it. Again, I'm going to stop sharing. Kelsey, if you can start again, and we'll pop the URL and code back up, so everyone can see it. There we go. Thank you. So again, take a picture, put the word out. Teachers, share it with other teachers, students, tell your friends. We really want to get some eyeballs on this. And as you can see, it's a pretty valuable thing you guys that we're giving access to. Spend some time. I think it'll help you as you work toward test day. I think that's it. That's it from me. Kelsey, did you, I don't see any questions. Maybe if we want to see if there's any final questions, you can post in the chat or in the Q&A. I guess give that a couple seconds. Kelsey, any final thoughts? Did you have anything you wanted to close with today?
Nope. So we just wanted to say thank you for your time. And the best thing to do is remember to keep calm and test on. And oh, there's a question and answer, I think. Oh, if we're taking the test, can we follow? Oh, okay, we already answered that one. Mark answered that in the thing about a 8-week plan, which you're getting close. It'll be tight, but you got to adjust as the weeks go by and adjust off a bit. But anyways, we want to thank you. If you have any further questions, we will stay on your past this recording if you have any questions. But otherwise, thank you for your time. Mark, do you have anything? Say?
No, I reiterate everything you said, and I just think practice is the key. The more familiar you are with the format, it's just another day at the office when you take the test then.
Yeah.
And so you know your strategy, you know what to expect, you know what's coming, and it's just working your way through it and then getting out of there.
Yeah, and having a plan and not trying to cram the last, like cramming the night before is not going to do the trick. You got to kind of go back and practice, and ultimately stay calm and test on. So anyways, thank you so much. We appreciate your time.
Last reminder to everybody, just that bit.ly, that URL you see on the screen now, take a picture. That's where the recording is going to live starting tomorrow. So anyone you know, tell your friends, and that's where it's going to be. So thank you, everybody. Thank you, Mark. Thank you, Kelsey. You guys did a terrific job. And good luck, everybody. Take care.